In this lecture, we're going to talk about some of the basic terminology that we use in host pathogen interactions. It's really host pathogen genetics, okay? Host pathogen interactions. So, going over some of the terminology, if we can't have standard terminology, standard terms that we use, it's just incredibly confusing, all right? I'll go through what is resistance, what is pathogenicity, and I'll talk about the gene-for-gene hypothesis. Most of this lecture will be on the gene-for-gene hypothesis, and I don't want to go too quickly on this because I think we do need to go through these concepts at a good pace so that you can learn. We are running a little bit behind time, so I might need to stop, and I don't know whether I'm able to continue after lunch, Gordon, for a little while. We have the country reports, but as I said, I don't want to rush this, okay? And I want to take my time and go through these concepts slowly. How many of you have actually heard of the gene-for-gene hypothesis? Can you put your hand up, please? All right. This is really the basis, it's the genetic basis of all of the work we do with breeding for resistance. You might not realise that, but I've talked about race analysis. Race analysis, identifying races or pathotypes, we are using the gene-for-gene hypothesis to do that, right? And when we're finding new sources of resistance like the ones that Gordon mentioned, the new resistance genes, again, we use the gene-for-gene hypothesis to do that. So it forms the basis of pretty much all the work we do. Very simple concept, but it has very far-reaching implications. Okay, so I showed this image yesterday. It's 2 wheat leaves, 2 wheat leaf rust pustules. This one and this one. What are we actually looking at here? How would we describe this one? And how would we describe this one? You might say resistance or susceptibility. All right, resistance here, susceptibility here. But that is if you're a plant breeder. That's fine. We'd say resistance and susceptibility. They are the alternate allelic states, if you like, of a trait we call disease reaction. And that's very much a breeder's perspective. But I'm a plant pathologist. I like the pathogen. So what would I call them? I'd say the one on the left is avirulent, and the one on the right is virulent. Okay, so that's the pathologist's view, the person that's working with the pathogen. We talk about avirulence and virulence. And they are the alternate allelic states of a trait in the pathogen we refer to as pathogenicity. Now I know that there's a little bit of confusion there with the literature. If you read some literature, the word pathogenicity is used differently. But in genetics of host pathogen interactions, the traditional way that we've used the word pathogenicity is to describe the trait in the pathogen that can either be, the state can either be avirulence or virulence. All right? Now the reality is, and I hope this shows on the screen, the reality is when we're looking at these leaves, right, we're not looking at a plant, we're not looking at a pathogen, we're looking at a plant and a pathogen. And not only that, we're looking at the outcome of the interaction between a plant and a pathogen. That's what we're really looking at there. It's the outcome of the interaction between two organisms. And this is what I mean by host pathogen interactions, or the genetics of host pathogen interactions. We're looking at the genetics underlying that interaction. And when we're talking about that, we would call the one on the left an incompatible interaction. Sometimes we say this is a low infection type. The one on the right we call a compatible interaction. The outcome is compatibility. We call this a high infection type. So the trait there is either the phenotype, right? I think you'd be familiar with the terms phenotype and genotype, right? The genotype is the actual genes that are present. The phenotype is what we see, the outcome of those genes. So we can call this an incompatible or compatible phenotype. Often we just say a low infection type or a high infection type. Now, one thing I'm going to do in my talk is in what I present, I'm going to refer to these terms down here. I'm going to show them in a red font, right? So you can, the reason I'm using red is you can think of this like a traffic light. It means stop. So where we have resistance or avirulence or incompatibility, stop. The pathogen is stopped. So red like a red stoplight, okay? Where we have susceptibility, virulence or compatibility, I'm going to use a green font. This is like go. The pathogen can go. So just think in terms of traffic lights, okay? If you get a little confused, if the font is red, we're talking about the terms in the left-hand column. If it's green, we're talking about the terms in the right-hand column. How many people have seen a picture like this? The different infection types that we have to rusts. Okay, you'll see this if you read the literature on rust where people are reporting on either rust isolates or resistance, and they're talking about seedling reaction. They use infection types on what we call a 0 to 4 infection type scale. All of these here we would say are low infection types, right? Or incompatibility, or avirulent pathogen isolate, resistant host. This one here, the 4 infection type, is high infection type or compatibility. Virulent, pathogen isolate, susceptible host. Compatible interaction.